Hello and welcome to video 4 from chapter 4, where we will just look at a couple of slides and talk about how behavioral genetics research is conducted. So the goal of behavioral genetics research is to establish the level of heritability, which is a measure of the extent to which genes determine a particular behavior or characteristic. So how much are your genes responsible for that behavior or characteristic or trait compared to your environment, your nurturance? So the research is designed to describe the genetic basis for behaviors, how much are genetics responsible or not responsible for behaviors and traits, and then some of the things that they study is things like alcoholism. Is it, how much is it your genes versus your environment? Depression, intelligence, personality traits, also certain abilities, like they might study musical abilities. Is it because you had a lot of lessons, or is it because you're born with musical genes? I mean, mathematics abilities have been studied as well. I mean, so there's a wide range of different behaviors and traits and characteristics that they study in behavioral genetics research. So how is this research conducted? Well, there's a couple of major ways that they do this. One thing I'm going to start with isn't listed here, but because it's kind of the least important, but they do what's called family studies, where they just study people that are related. So they're not adopted, they're not twins or anything special. I mean, there's, like how similar are two siblings to one another? How similar are they to their parents and to their, and they might study cousins and uncles and aunts, and how similar to your grandparents. And they might, so in family studies, they look at a lot of members of a family, and they try to draw some conclusions based upon how certain behaviors or traits are passed on, and do they, and which members do they appear in more or less. Anyways, those are called family studies. More relevant, a little bit more relevant, are adopted children studies. And particularly when the children are adopted from birth. Those are the studies you really want to do when the biological parents did not do any raising of the children. So what you end up with is when you look at children that have been adopted, I mean, their biological parents contributed all their genetics, but none of their nurturance. They didn't make decisions about how to raise these children or have any influence on that. Their adopted parents did all the nurturance, and, they raised the kids, but they have no genetics in common. And so it's kind of like you're pitting, are these children more similar to their biological parents because of their genes, or are they more similar to their adopted parents because those are the people that raised them? And so they calculate what's called concordance rates. A concordance rate just means degree of similarity. So how similar are the adopted children to their biological parents? And they can come up with a number, for a certain trait or behavior, and how similar are they to their adopted parents? And so these are somewhat and similar to their biological parents that, they may not have raised them at all. So all those years of raising children, I mean, you certainly have influence, but the parents that just contributed the sperm and the egg, and often children are more similar to them because of their shared genetics. Okay. Even more relevant are twin studies. This is kind of the gold standard of behavioral genetics. You get more information from twin studies than either adopted or family studies. This is where, the real serious research gets done. And many people right away imagine that the typical case is studying adopted, no, not doctors, excuse me, studying identical twins that have been separated at birth. And we will look at that in a moment, but that is not the standard study. Okay, that's an interesting case when children have been split right from birth and grown up in different environments where they share genetics and then you might see them as adults and see how similar they are, even though they didn't grow up together. And we will look at a video of a case like that in a moment. But the more typical and more powerful study is that they compare pairs of identical twins and they look at how similar they are on a behavior or trait, and they compare it to pairs of non-identical dizotic twins, meaning fraternal twins, twins that do not look similar. They may not even be similar gender, the same gender. So let me get into the logic of this, and it all revolves around concordance rates, degrees of similarity. Now, let me first go to a whiteboard here and talk a little bit more about identical versus non-identical twins. And where is it? Oh, here. No. Give me one moment. I'm missing my whiteboard. Oh, okay. I know where it is. Okay. Sorry. We're getting there. Just give me one moment. Here it is. Okay. So I just made a couple of notes here. So just to make sure it's clear, a lot of people aren't necessarily aware of, how identical versus non-identical twins are formed. So let's start with the identical twins, or MZ twins. MZ stands for monozygotic. Mono means one. Remember, zygote is the first fertilized cell where the sperm meets an egg and creates a zygote. So monozygotic means that identical twins come from one zygote. There was one sperm and one egg that led to these identical twins. So what happens is that after a fertilized egg begins to divide and multiply, which is normal procedure, one cell becomes two, two become four, four become eight. And we will talk about this in the next chapter. But at some point along that sequence of the cells dividing and multiplying, sometimes that ball of cells splits in two. And so each of those cells is an exact duplicate of that first zygote. So that zygote, that one zygote becomes two identical zygotes, or they're not called zygotes anymore, but two identical cells. And those two cells become four identical cells. So when the ball of cells splits into two, you have now two balls of cells that are genetically identical, 100% similar genetics, exact match, okay? Because each of the cells in each ball have exactly the same genetic material. This is why identical twins, look exactly alike and have similar characteristics. They start with exactly the same genes. In the case of non-identical DZ twins, DZ meaning dizygotic, di means two. So this is when there are two separate ova or eggs that are fertilized by two separate sperm. So usually in a normal menstrual cycle, a woman will release one egg per cycle, but sometimes there are special cases or complications where more than one egg may be released. And in particular, there are cases where women might be going through in vitro fertilization, where they have multiple eggs implanted into the uterus in the hopes of one or more of them developing into an embryo. And so there are both medical cases where it happens, and there are drugs that can be taken by women that may cause them to release more than one egg at a time. Anyway, so two separate eggs are fertilized by two separate sperms. So these twins are no more genetically similar than any two siblings, even though siblings are born years apart. The degree of similarity of these two twins developing at the same time is the same as any two siblings. Like I said, they might not even be the same gender. One may be male, one may be female. They're not going to look alike. They may have very different characteristics or traits, just like brothers and sisters do. So they're approximately 50% genetically similar. It doesn't have to be exactly 50, but it's close to 50% genetically similar. Okay, so we have these two types of twins. The logic of a twin study is that you take a pair of identical twins and you see how similar they are on a certain trait that you're interested in. So maybe I'm interested in IQ, and I see how similar. I collect a bunch of identical twin pairs, and in each pair I see how similar mathematically are their IQs. And then I gather a bunch of non-identical dizygotic twins, or also known as fraternal twins. I forgot to write that down. They're called fraternal twins when they're non-identical. And we look at their degree of similarity. We can calculate concordance rates for both the identical and non-identical twins on a certain trait. Now, if that trait that we're studying is caused much more by genetics, then the identical twins who are exactly the same genetically, they should be a lot more similar on that trait than the fraternal non-identical twins. If, however, it's something that's caused more by nurture, by environment, then we don't expect the identical twins to necessarily be more similar than the non-identical twins. In both cases, the identical twins, we're looking at normal households where they're growing up in the same household with the same parents, and so they have a lot of similar experiences, but so do the non-identical twins. They grew up together, they're the same age, they're in the same household with the same parents, and so the idea is that if genes have nothing to do with something, then both sets should be close in their concordance rates. I'm going to show you an example to really kind of help nail this idea home. Let me go to a new share now, another whiteboard. Okay, and these are real results from two twin studies that were done. One of them, the first one, was looking at autism, or autism spectrum disorder, ASD, and they calculated concordance rates among the identical twins. And this figure, the concordance rate is going to be between 0 and 100%. 100% would mean that if one of them has autism, then the other one always has it. That would mean it must be completely genetics, because and that's what would lead to a 100% rate. If there was 0%, it would never be 0, but there's always going to be some percentage. But if it's close to 0, that means that probably that if one of them has it, it's not very likely that the other one also has it. Anyways, for identical twins, look at the figure of autism. It's 95.2%. So if one of the twins has autism, 95% of the time, the other twin also has it. Wow, it's very close to 100. That means there must be some genetics at play here, but we don't really know that until we look also at the fraternal number. And you compare the two numbers. This is the key, is the comparison of the two numbers. For the fraternal twins, it's 4.3%. If one of them has autism, it's only about 4% chance that the other one also has autism, very low rate. When there is a large difference between these two numbers, that points towards genetics being at work. When the identical twins their concordance rate is so much greater than the fraternal twins, that means genetics are at work. And that's why they're showing so much more similarity. So we see a huge gap there between the two figures. This is a kind of extreme case. Autism is almost all genetics. It's like I said, that number is close to 100%. Let's compare it to another result, secure attachment. In the study, they looked at if one of the twins is securely attached to the mother, what's the chance that the other twin also has secure attachment to the mother? And we will study secure versus insecure attachment later in the textbook. But anyways, look at the numbers here. Very different pattern. The identical twins are at 70% concordance rate. If one of them is securely attached, 70% likely that the other one is. The fraternal twins number is just slightly lower, 64%. If one of them is securely attached, the other one is 64% likely to also be securely attached. When the numbers are close like this, and these are very close numbers, and usually we expect, you know, if there's any genetics involved at all, then the identical twin numbers should be somewhat higher than the fraternal twins. So, but these numbers are really close. What this tells us is that secure attachment is not carried through your genes. Maybe a tiny sliver, which might lead to that 6% difference, but that's probably not even statistically significantly different. But anyways, the numbers are so close that what this tells us is that secure attachment is gained through nurturance. It is gained through the mother's actions with the particular infant child and how the mother treats them and how they're there for them. It's not, you're not born securely attached or insecurely attached. It's really dependent on the mother's sensitivity and caregiving towards each child. And other research backs this up. We really don't. We really see that it is a environmental influence. Whether you become securely attached or not is dependent on situation and the mother's caring for you. So when the numbers are close, it points towards environment, nurturance. When the numbers are far apart, it points towards genetics and genetic influence. And this is the main type of study that is done in behavioral genetics. They gain a lot of information from these twin comparisons, comparing identical twin concordance rates to fraternal twins. Okay. All right, hopefully that all makes sense. I'm going to go back to the PowerPoint. Yes. Okay, here we go. Okay. Now I told you that there's also the case of studying identical twins reared apart. This is an interesting type of study, but once again, by no means is it the standard. It does not give you as much information as what I just showed you, the study of identical versus non-identical twins. But let's look at one case study example. I'm going to show you a short video where they are studying two women that were separated early in life and have now been reunited as adults. They are identical twins that were separated. It's kind of interesting. Let's take a look. In one moment, I click a couple of boxes. Okay. And where are we going? Here. Okay. I will be there. Okay, sorry, here, click a few things. Here we are. So that's a look at a particular study of identical twins separated and raised apart and reunited as adults. Okay, just to finish off this section, just one more note. What about triplets? Are they more likely to be monozygote? Are they going to be monozygotic, or are they going to be non-identical or identical? I mean, like, it turns out that every combination is possible. You could, if you had triplets, they could all be identical, where you started with, once again, a single zygote, and it split twice, creating three separate individuals. They could all be non-identical. In this case, it wouldn't be called dizygotic because that means two, but I guess it'd be called trizygotic. I'm not sure, but anyways, it could be three separate eggs fertilized by three separate sperm, and so in that case, you know, it might get two boys and a girl, two girls and a boy, whatever. You can also have a mixed case where you, in a set of triplets, you may have two of them that are identical and one that's non-identical. So that would happen where you start off with dizygotic twins. You have two separate eggs that are fertilized by two sperm, and then during the dividing and multiplying, one of the sets splits apart, creating identical twins. And so there's any kinds of combinations possible. If you had four babies, I mean, you could have two sets of identical twins, or they could be all identical, or all non-identical. Okay. Anyways, that concludes this section on behavioral genetics and how we study it with adoption and twin studies. In the last video, we will look at how the environment and our genes interact. Oops. Okay, and I will talk to you next video.